Group problems for section 1. Question 1 is defining some terms. A gene, a segment of DNA that functions to produce an RNA molecule. Chromosome set, all the chromosomes inherited from one parent. Human cells have two sets, a maternal and a paternal. Homologous chromosome is a set of one maternal and one paternal chromosomes. Sister chromatid, it's a pair of replicated chromosomes that are attached at the centromere. Their sister chromatids are genetically identical. An allele is an alternative form of a specific gene. Homozygous and heterozygous. Homozygous is a diploid individual that has two identical alleles for a particular gene, while heterozygous is a diploid individual that has two different alleles, two different versions. Dominant and recessive. Dominant describes an allele that determines the phenotype in a heterozygous condition, while recessive is a trait or a gene that is masked by if the dominant gene is present. And then genotype and phenotype. The genotype is the genetic composition, while the phenotype is what we see, the observable traits. Questions 2 and 3, you're asked to produce drawings of illustrating meiosis 1 and meiosis 2. I found these to be useful. In this illustration, it doesn't show the substages of prophase 1, but there are 5 substages, but the 2 that we're asked to key in on are zygotene and pachytene. The important part of zygotene is where synapses occurs, while pachytene is where crossover initiates. And the way I remember that step is Manny Pacquiao is a boxer, and he's a southpaw, so he has a left cross, so crossing over. During metaphase, know that they align. Anaphase, they separate, and then telophase is where they completely become two cells. Notice that the nuclear envelope begins to reform, and they're diploid cells. Important to note, because in meiosis 2, the ultimate outcome are haploid daughter cells. But similar steps. The nuclear envelope begins to break. They align. They separate. And they become ultimately 4 haploid daughter cells. In question 4, you're told that in chimpanzees the diploid chromosome number is 48, and then you're asked to identify the total number of chromosomes, number of homologous chromosomes, and number of sister chromatids in each of these phases. So for these problems, I like to set it up this way. Just make a chart or a graph or whatever you want to call this. The stages, and then what you're asked to identify, and this will vary over time, different questions. So prior to DNA replication, the beginning of metaphase 1, those two got bunched up together. So prior to DNA replication, so what I like to do is bring up that image to answer these questions. So prior to DNA replication, there are 48 total number of chromosomes, 24 homologous chromosome pairs, and 0 sister chromatids. However, at the beginning of metaphase 1, you have 96, or double your number total chromosomes, you'll have 96 sister chromatids. So I've added the answers. If you look here at the end of telophase 1, we're here. We're still diploid. So we have 24, the total number of chromosomes. We have 48 in your number of sister chromatids. At the end of telophase 1, since we're haploid, we don't have any number of sister chromatids or homologous chromosome pairs. After fertilization, essentially it's like saying prior to DNA replication. But after fertilization, we'll have again the 48, the 24, and the 0. Question 5 says, you suppose you are examining plant height in pea plants, and plant height is controlled by these two alleles. And so if you have a cross between a heterozygous tall pea plant and a dwarf plant, predict the genotype and phenotype. So I've already set that up. Let me pull up that document. So I made this square. Let's see, we have a heterozygous tall and a dwarf. Since the dwarf is recessive, we know the genotype would be TT, or lowercase t, and heterozygous tall will be T uppercase, lowercase. So when you do the cross, you have these 4 results here. So TT 50% of the time, and lowercase t 50% of the time. So, in terms of genotypes, as I mentioned above, and phenotypes in this case will also be 50%, 50%. This should be auto-corrected. This should be TT. They give a more complex example, so I pulled that up. Let me pull that up. It says here, let's consider the following characteristics. So we have pod texture and seed color. So a cross is made between a pea plant that has constricted pods and is heterozygous for seed color, and a plant that is heterozygous for both. Assume these two assort independently. So again, I've set that up. Let me pull that up. So I've copied here the 2 crosses we're making. A plant that has constricted pods and is heterozygous for seed color. So since constricted is recessive, we know it has to be 2 lowercase, and heterozygous seed color, that means uppercase, lowercase. And a plant that is heterozygous for both, so uppercase, lowercase, uppercase, lowercase. To solve this one, you can do the FOIL method. So you'll do this lowercase s, and you'll cross it with the first s, so uppercase, and then again first s here, cross with the second. So in essence, you're doing s when you cross it the first time, and you cross it, and it's going to do autocorrect the second time. And you take the second allele here, s, the lowercase s, and you cross it again, and one last time. And then you do the same with the y, which I've done down here to save time. And you're given these fractions. So half the time when you do the cross, it's smooth. Half the time it's constricted. And over time you'll see that whenever they're heterozygous, you have 2 heterozygous, you always have a 1/4, 1/2, 1/4 outcome. So the question, what did it ask? It asked to determine the genotype and the phenotype. So there are 4 possible phenotype outcomes: the smooth yellow, smooth green, constricted yellow, constricted green. To solve this, what you do is we find possible smooth, which is 1/2 of the time, and you multiply that by the possible yellow, which is 3/4 the time, not 1/2 and not 1/4. You have to add those, so 3/4, which will equal 3/8. Again, with the smooth green, you're 1/2. We know that, and green will be 1/4, which will give you 1/8. Alright, to save time, I did the remainder. So we have constricted 1/2 the time, yellow 3/4 the times. So I'll do the phenotype, and then the constricted green 1/2 times 1/4. That gives you 1/8. Lastly, you're given this theoretical situation where you perform a cross between this genotype and this genotype. And importantly, though, we're only asked to find out the proportion of the offspring expected to have this genotype. So similarly, you could do the FOIL method, where you would cross each one, and you would get these results here. So you do uppercase A with the lowercase, uppercase with the lowercase again, then lowercase with the first lowercase, and so forth. Same with the B's, uppercase, uppercase B, uppercase, lowercase, etc. So I went ahead and did those crosses, and you're given these fractions. I'm trying to be brief, and I can't believe already 12 minutes have passed. So to find out the expected proportion to have this genotype, all we would do lastly is multiply these fractions, and when you do so, it equals 1/64, or which is 1.6%. I don't remember. You know, have to do that. Let's see really fast. Uh, 1.6, yeah. 1.6% of the offspring are expected to have that genotype. Let's see. I hope that's clear enough.